Stop when you finish, and the floor is yours.
Thank you very much, and I'm very happy to be here with you on the screen and sharing our research and our research interests. And the title of my presentation is Effects of Saltation and Abraded Silicates on the Survival of Bacteria and as a Sink of Methane and Carbon Dioxide. And I will start out with an iconic picture, and some of you may have seen it. It's not a picture, of course, it's a drawing, and it shows the Viking lander, one of the Viking landers on the Martian surface. And this happened almost 40 years ago. It was the first mission to Mars of a lander, 2 landers, 2 satellites, and it all worked well. And at that time, people believed that there was life on Mars. I mean, some of us may still do it. And they constructed an instrument that should prove it. So it was sent to Mars, it landed on Mars. And if you think about the technology that people had at hand at the time, it was extremely courageous and almost a miracle that they were so successful. They did 3 major experiments where they were looking for the possibility of life on Mars. And I just have a very primitive sketch that summarizes these 3 experiments. One was dealing with the possibility of photosynthesis, one was dealing with the possibility of heterotrophic processes, and there was one, which is the one in the middle. Can you see my mouse? Can you see that?
Yes, we can.
Okay, this is the one that I want to talk about a bit. This is the one in the middle, and it's a very enigmatic experiment. They took some Martian regolith surface material, put it in a container, exchanged the air with the Martian atmosphere, and then they were adding a solution of different kinds of organic material that was radio labeled. And the idea was that known from Earth and from experiments that microbiologists did at the time, if you would see a production of CO2, that would be due to the presence and activity of microorganisms. So the idea was then to add these a series of different compounds. It included sugars, amino acids, fatty acids, all kinds of stuff that microbes on Earth appreciate, like to digest and to produce CO2 from. And then this CO2 was led to a radioactivity detector, a Geiger counter type of instrument that could measure the radioactivity of the released CO2. And here is the experiment, and this is original data from one of these Viking experiments. And what you see here is on the x-axis, you have the time in Sol. Of course, this is Mars, this is not the Earth, so rather than counting days, you count sols. And 7 sols is approximately a week on Earth, also on Mars. And then on the y-axis, you have the radioactivity in counts per minute. So what you see here is first the addition of the injection of the radio labeled compounds, and then relatively short after the injection, you see an increase in radioactivity. And if you had done this experiment with just some garden soil, you would see the same here. You would say, okay, this is a clear sign of biological activity. They also did a control experiment, which is the curve here at the bottom, and it was an excellent control. They heated up the soil to 160 degrees. And we know now from Earth that the highest biological activity that has been measured was measured up to 120 degrees, so far above. This one was above this temperature, so everything microbial should be killed. And you see here, the response is just as you would expect, no release of radioactive CO2, or only very, very little compared to your experiment. And then they, after some days or after some sol, there was no more release of radio label, and that could just be that these microbes in the Martian regolith, they didn't eat all the different compounds, but they were specialized to only eat a few of them. So what they did, they injected a new batch of label of the same composition, and the result was a bit disappointing. So they saw that the radioactivity actually went down. This could be explained by the absorption of radio-labeled CO2 by the liquid that was added, but there was no further release. And this has been debated since. Now it's almost agreed that this is probably not due to biology, but rather to chemical activity. This is something that has interest, that we are very interested in, is figuring out, okay, what is the reason for this Martian regolith reactivity? When the people in our group don't think that this is biology that can explain these results, but it's due to the fact that something in the soil oxidizes the material and releases CO2 that then could be measured by the Viking. So we think that the experiment worked perfectly well. Just the cause or the reason why CO2 was released was not biology, microorganism, or whatever organism, but it was due to a chemical reaction. So what could be the cause of reactivity? There are several ideas. These are ideas that are not put forward by us, but by others. And the cause of reactivity that was suggested could be hydrogen peroxide. And the way how hydrogen peroxide could be produced is there are several alternative explanations that could take place altogether. One is photochemical reactions in the atmosphere. You know that there is water on Mars, and there's also water vapor in the atmosphere, and that could be oxidized due to the high radiation, UV radiation, cosmic rays. Hydrogen peroxide could be produced, and over the many million years, this hydrogen peroxide could diffuse into the soil or regolith and then accumulate there. Another possibility is triboelectric charging in dust devils. That means when dust devils are produced, the particles in the dust devils, they collide with each other, bump into each other, and that leads to charging. And then when the dust settles again, this charge could also be a cause of reactivity. Alternative, it's mineral surface that are activated by radiation or meteorite impacts, as you see it here on this figure. And for this process, the meteorite impacts would just fracture the the regolith. New surfaces would be produced, and these surfaces could be the cause of reactivity. So the hypothesis that we work under is that abrasion, and I will just tell you in a bit what abrasion is. By saltation is a mechanism of soil reactivity production. And one of the types of reactivity could be reactive oxygen species. And these reactive oxygen species, they include hydrogen peroxide, but also hydroxyl radicals. So when we look on Mars, Mars has an atmosphere. It's not comparable to Earth's atmosphere. It's only 8 millibar when it is the highest, but still enough to move particles at the surface. And there are different ways of how they can be moved depending on the size and the weight of the mass that they have. If it's fine dust, it can be suspended and enter the atmosphere up to higher latitudes. If it's larger grains, they would just jump over the surface, and this process is called saltation. And what this process would lead to is when the sand grain hits the ground, hits another sand grain, it gets fractures. And this fracturing of a sand grain is what is called abrasion. That's the last process is creeping. That means that bigger particles just roll along the surface and then could also be eroded during that process. And that erosion actually takes place on Mars, and that these processes are processes that takes place on Mars. You can see on the pictures here. This is pictures of Mars taken at 2 different points in time. Here you have a clear view on the Martian surface, and here this is Mars covered by global dust storms. And these global dust storms take place quite frequently. And in these dust storms, particles or dust can collide. It can be charged by triboelectric charging and then be activated. Another phenomenon that has been observed frequently, I think this is a picture taken by this Spirit rover. This is a dust devil just passing by the rover, and you can see that dust is levitated here, and you can see all kinds of, can have all kinds of processes taking place between the particles. Another example of wind effects are dunes, and they only form when there's wind present. And there are pictures, so many pictures taken of the Martian surface that you can actually follow specific dunes, and you can see that these ripples are moving. So even larger particles of sand are moved And another example are these surfaces, and these surfaces can only be produced if you have some kind of erosion going on. So we are interested in the effect of abrasion products. This means fractured silicates, minerals, basalts, and also the effect of the abrasion process as such. And there are two major foci that we have. One is the survival of microorganisms. This is the first part of my presentation, and then I will talk about the composition of the atmosphere and how that can be affected by abrasion. So this is our experimental setup. Of course, I mean, it's difficult to simulate Mars on Earth. I mean, we actually have a wind tunnel at our university, which is quite close to the Martian situation, but this wind tunnel is very big and is also very costly to operate. So we have created our own Martian atmosphere. We use these ampules that you can see here. They're about 20 centimeters long, and then we can fill them with different kinds of material. In this case, it was basalt, and here it is quartz. And then we exchange the atmosphere. We close them at the end. So we have an enclosed system with different kinds of minerals in a Martian atmosphere. The way how we produce wind is by putting them into this box and then turning the wheel. And we turn the wheel at about 30 turns per minute. And this is approximately simulating the highest wind speeds measured on Mars, which is about 15 meters per second. If we do biological experiments and we're interested in the effect of the eroded material on microorganisms, we grow the microorganisms. I will just show you in a minute which types we have tested, grow them in a medium. Then we harvest the microorganisms, wash them in a buffer, determine the number, the concentration, and then titrate them to the proper concentration. And then what we do is we take our eroded material, as you see here, we add the microbes, and then we evaluate the survival by taking out the microbial suspension from here and then plate it and count the colonies, multiplying it by its dilution, and then we can get the total number of surviving bacterial cells in this situation. So we used three different model organisms. One is Pseudomonas putida. It's a common microbe in soil, and we use this because soil is in principle, each soil is an extreme environment. During the summer, you can have high temperatures. During the winter, you have low temperatures. You can have water saturated situations. You can have situations where there's very little water. So these organisms have to cope with all these changes to keep their homeostasis. Then we have Deinococcus radiodurans. And for most of you who work in the field of biology, you know that this organism has been used as a model organism in astrobiology due to its radiation tolerance and also its desiccation tolerance. And then last but not least, these are gram-negative bacteria. Deinococcus is a bit special with its cell wall, but still it counts as a gram-negative one. And this is a gram-positive one, a spore-forming organism, Bacillus subtilis, which has been used in many different studies, particularly because of its ability to form spores. And these spores are very resistant to all kinds of stress. So, and now I show you only a single example of the results that we got, and this was with Deinococcus radiodurans. Here you can see two different graphs on the left side of the slide. This one was Deinococcus radiodurans cultured. These cells were harvested in exponential phase, and this were Deinococcus radiodurans cells harvested in stationary phase. And for those of you who are not biologists, so if you follow the growth of a bacterium, you can see that they have a phase where they multiply very fast, and so some of them multiply so fast that they have exponential doubling. And in the stationary phase, you still have living cells and also growing cells, but you don't see a change in cell number anymore. The cell number in the culture stays constant. And these cells, even though it's the same type of cell, they are physiologically different. So it's important to see how cells behave that are exponentially growing, very active cells, and these cells are less active. And you can see that just from the figure that there are some differences. Just to explain the colors, we did these experiments with two different types of minerals. The blue ones are with basalt, and the orange ones are with quartz. And you see already just from the color, you see a difference that only with basalt we saw an effect. And you can see here this is the percentage of surviving cells normalized to the beginning of to a culture that was not subjected to the eroded material. And you can see with quartz, for example, there was no change in number of cells. You can even see that the number of cells increased, and this is not because there were nutrients added to the buffer, but it was simply because these cells have storage compounds that they activate, and then they can multiply even though they don't have external nutrient sources. You can also see that that for the blue ones, there's one curve that has a steep drop in the beginning, and one that stays constant over the whole experiment. And the experiment lasted for about a day, and this means that the cells were subjected to abraded material for 1 day. The upper one, the blue one, is our control, and this is the control was made in a way that we had these ampules with quartz or basalt, and then we could open them either in a glove box where there was no oxygen present, or we could open them in the lab where there was oxygen present. And we observed that when we opened these ampules in the presence of oxygen and the abraded material was subjected to oxygen, the reactivity was eradicated, so it stopped. So we only see an effect, and the effect in this case is the killing of the bacterial cells when we opened these ampules and subjected the bacteria under anoxic conditions, when there was no oxygen present. You can see that there's a very steep decline in the number of cells, and already after an hour, we are at the detection limit of our method. And this method is really sensitive. We start out with 1 million cells per milliliter, and here we have no cells left already after an hour, and this was with cells that taken from an exponentially grown culture. If we take a stationary phase culture, you can see that the pattern is different. At the end, after 24 hours, we are reached the same. The asterisk, the star, explains that this is the detection limit. So here we could not see any more cells. So it was at least below 0.1 per mil. So very few, if at all, very few cells, but you can see that the cells are more resistant. They die slower, so to say, but in the end, after 24 hours subjected to this material, they were all dead. That means that our artificial Martian regolith has a very, very strong effect in terms of killing cells, even the cells of Deinococcus radiodurans, which is famous for its resistance to both desiccation and radiation. So, but apparently subjected to our abraded basalt, the cells die, and they die quite quick. And this, of course, has some implications for forward contamination of the Martian surface, which could be positive. So it seems that cells, when they get in contact with water, die fast, considering that the Martian regolith is activated in the same way as in our experiments. Then we did an experiment where we took the spores of Bacillus subtilis, and again, the same experimental design, but now rather than subjecting the cells to abraded quartz, we took the cells, we spiked our material with, and in this case, it was only quartz. We spiked them with the spores. We took the ampules that were with spores and quartz into our mass wind simulator, and then took ampules out after a certain amount of time and determined the number of surviving spores. And here you can see the result. Already after a very short time, after 5 minutes in our under abrasion, you see that 50% of the spores were killed. And this is a power log function. You can see after 100 hours, there was less than 1 per mil of the spores left. So apparently this physical abrasion process is very tough to the spores and kills them very efficiently. This is a result that we published in Astrobiology 2 years ago. And this year we had a very interesting discussion with some colleagues, and they wrote a comment on our study that was published in Astrobiology, and we wrote a reply to that comment. It was by Mintz and al., so you could find it here. And they criticized our experiment, saying the material that we used is not comparable to the Martian material because our material still has sharp edges, while we expect that Martian material has rounded edges. And that was also one of the arguments that we used to explain our results. So in order to test our system, what we should do is to repeat some of the experiments using sand or minerals that don't have sharp edges, but that are already treated by abrasion. That means that the edges have rounded. I'm still convinced that we will see the same effect, that we also will see that the spores will be destroyed by the impact of the abrasion process, but just by the saltation process, not necessarily by the effect that the material is broken up. And then we will see that the spores disappear. But of course, we have to redo the experiment under conditions as they have been suggested by Mintz. It was a very interesting and very nice discussion that we had on that problem. This was done in the same. We were interested in to see whether the atmosphere and the atmospheric composition had an effect, or if it was the mechanical effect that eradicated the spores. So here, this was our control experiment, 8 millibar stationary control, and here we run it under different conditions for a couple of just a day, and you can see that the atmosphere and the pressure of the atmosphere didn't really matter. At 500 millibar, we saw the same as at almost vacuum, or as close to vacuum that we can produce in our system. So it was the mechanical effect and not the effect of the atmosphere that destroyed the spores. So here's our conclusions is that eroded silicates, and we think that it's an overlooked source of toxicity, which has implication for Mars habitability, but also for contamination. Silicates produce reactive oxygen species. We actually measured that. So we could both measure hydrogen peroxide and hydroxyl radicals when they get suspended in water. The production of these measurements depends on the type of silicate and the composition of the atmosphere. The toxicity may be due to these hydroxyl radicals produced due to Fenton chemistry, and the abrasion in itself may be an effect, maybe have an effect that destroy chemical structures, including endospores. And here I'll just remind you of this conversation that we had with Mintz and their group. So maybe we should look a bit more into detail how these minerals should look like in this to see to evaluate their effect. Research question is, so what kills the cells? We didn't really evaluate what actually was detrimental to the cells. We have an idea that it could be lipid peroxidation due to the formation of these oxygen radicals, so that the membrane is actually destroyed. We also should look more into the role of different the particles. We always had a specific size of particles. We never tried these very fine dust, as it is present on Mars and participates in the dust storms. We need to look more into the understanding of this abrasion chemistry, and also the direct effects. Test not only organisms, but also look into whether it could change organic molecules, and could be an explanation for why there is so little organic material on the Martian surface, even though we have these micrometeorites that have delivered lots of different kinds of molecules and a lot of organic material over the last billion years. So next part is abrasion of silicates and the composition of the Martian atmosphere. And this was stimulated by the observation of methane in the Martian atmosphere. This happens for the first time about 20 years ago. And you could also see here that Martian methane is not distributed evenly. You have hotspots, the red parts with high methane concentrations, and the blue parts with low methane concentrations. And this is interesting because it tells you that there is a release of methane. It's not so that it's just there, but it has a source, and it may even have a sink here at these sites where there are lower methane concentrations. And the interesting thing with methane is that there have been several studies of methane, and there have also been studies that showed that there was no methane in the atmosphere. So apparently there is some dynamics. It has some cyclic appearance and disappearance. And this is a figure from the Curiosity rover, where you can see that there are annual shifts in the concentration of methane. So this is measured over three Martian years. We were into it. The interesting thing is here that you cannot really explain these rapid changes, at least the rapid depletion of methane, by photochemical processes in the atmosphere. So the lifetime of methane would be too long for it to explain the change. And our idea was, okay, maybe it's not photochemistry, but maybe we can link that to this abrasion process that we have studied. So we were carrying out the same kinds of big experiments, the same setup, and here you see pictures taken from our ampules with different kinds of minerals. Here we have olivine basalt, we have plagioclase, we have augite, and this is quartz. And you can see that there's clearly different amounts of energy that are released when you move the sand. And here what we did, we just took an ampule, filled it with the material, and then we shook it and took pictures with a camera. And you could see that there is light emitted during that process. And the idea is here, okay, if we have such a high energy release, maybe this the energy that is released, and our idea is that you have triboelectric charging of the surfaces, charge separation, and then you have electrons that jump from the charged material onto the gases and then ionize the gas. It can go both ways, and in this case it was argon, and argon gives off electrons. The electrons are then taken up by the charged material, and you have ionized argon. And this is actually the case when we use quartz in a quartz ampule. What you see here are figures showing the black ones are the experimental. This is the wavelengths at which we measured with the spectrophotometer, and you see these different sharp peaks. And the black ones are what our experiments showed, and the blue ones is what you can get from the literature. And you can see that there's a very nice correspondence between what you would expect from literature values of ionized argon and what we see with our experiments. We can also see that not only argon is ionized, but also nitrogen. And our argument is then, okay, argon and nitrogen are very difficult to ionize. You need a lot of energy. So what about methane? And methane needs a much lower ionization energy than argon does. So maybe methane could be removed from the atmosphere by this process. And we did an experiment. We constructed an ampule like this. This is not the one that I'm showing here, but just to show you the experimental setup. We had our ampule. We put on a pressure gauge to measure the pressure because we thought this is maybe a more easy way to see whether our methane is removed. And it would also tell us if we have a leak. So if we have a leak, then we wouldn't see a decrease in pressure. And that's actually what we see here. We fill the tube with a certain amount of methane in the atmosphere, and you can see that after a relatively short amount of time, all the methane is removed by this process. So it could be that abrasion and the production of new surfaces leads to the ionization of methane, which then reacts with the mineral. And we did some experiments with quartz, where we saw that with NMR, and we used methane that was C13 methane, and with NMR we could see that the methane actually binds to the silicon surface. This is the control, and this is quartz that had been tumbled in a methane atmosphere. And when we tumbled that for a long time, and here the experiments lasted for almost a year, what we could see, I hope you can see the color here. This is quartz that was tumbled in the Martian atmosphere for about, and here we have an atmosphere that is exclusively methane. We can see that it was white to begin with, and now it has this brownish color. And what we did is we heated it first to 250 degrees, and you can see there's no change. And then we heated it up to 500 degrees, and you can see that the color disappeared. And we did some follow-up experiments, and you can find the data in this paper down here, that we actually have the formation of aromatic organic compounds that are detached to the silicon matrix. So not only removal of methane, but also synthesis of more complex organic molecules by that process, which is very interesting. So then we had we asked the question, okay, this was in the case this was basalt, and this was quartz, the experiment that I showed you before, and we also saw from the pictures before that there's much more energy released when we have quartz present. You saw that it was much brighter, much more red than the in color than the basalt. It was just only faint blue. So what is happening when we use basalt instead of quartz and tumble that in our quartz tube? And you can see also here after about 50 days, this started out with an atmosphere exclusively of methane, of 8 millibar of methane, and you can see that it gets removed, and after 50 days all the methane is gone. So it seems that also basalt can remove methane. But we have the problem here was that we had basalt in an atmosphere in a tube of quartz, and we were not sure what actually was responsible for the reaction, whether it was the basalt or whether it was the quartz. So we designed a new experiment, and for that we got some basalt from Iceland, and the basalt happened to have about the same composition as the basalt that has been analyzed on Mars. So we got a rock from Iceland. We drilled it, made an ampule. You can see it here, a kind of urn. We filled it with basalt, and then we put it in this container that we built. It took some time to get it tight, but now it's tight. And then we could both measure the pressure, and we had some windows here where we could spectrophotometrically, we could measure the gases. We could measure, and this was methane in a Martian atmosphere. So it was methane in an atmosphere that consisted mainly of CO2. And the result was quite interesting and surprising. So what we saw here, it's difficult to see there. This is just the axis. We have two different compounds. We have methane and we have CO2. So what you see here is that over a period of 70 days, our experiments last for quite some time, so you have to be patient. We saw that CO2 was removed, but methane was not removed. So that means our conclusion is that the removal of methane that we saw in the previous experiment was not due to the presence of basalt, but it was due to the presence of quartz. You can see here from the shape of the curve that it takes some time before the process starts. And we think that the basalt that we have added to the quartz tube erodes, removes the quartz from the tube, mixes it into the basalt, and then it's the quartz that actually is the reactive agent and removing the methane. But interesting enough, in this experiment we saw that basalt actually removes CO2, and this is known from Earth. When you break up basalt and you just expose it to a CO2 atmosphere, CO2 is absorbed. The question is then, okay, how can it be that there's still so much CO2 on the Martian atmosphere, considering that you should have such an efficient removal of CO2? What we are now going to investigate is when you look into the atmosphere of Mars, there's very little methane. It's in the ppb range, but there's still some oxygen, because oxygen is produced in the atmosphere due to the splitting of water in the atmosphere. And it could be so that it's not And we have observed that in another experiment, which I will just show you in a second, that it could be the small amounts of oxygen that are in the atmosphere that actually react with methane and remove the methane. And it's not the direct reaction of methane with the basalt that has been eroded. And this is the next experiment that we are going to carry out. So these are data that have not been published, and you are the first to see that. We did an experiment. So we went back to Earth. We were interested in seeing, okay, what would happen if we had an Earth atmosphere rather than a Martian atmosphere? And there we saw that in an Earth atmosphere, CO2, methane, and nitrous oxide are consumed when we erode during our erosion experiments. And this is interesting because all these three gases are greenhouse gases. So it's an interesting observation, and we're going to study that further because it could have implications for the atmospheric composition of greenhouse gases, and maybe it can also be used as a mechanism to remove greenhouse gases from the atmosphere. You know how urgent that is. So to end my talk, just a short summary for the last part. So the type of minerals decides on its reaction with the gaseous phase. We saw that clear. It depends on whether we have quartz or whether it's basalt that is used, and our results may have implication for the processes on Earth as well as I've shown you in the last picture. And with that, I'll thank you for your attention, and I'm happy to take questions.